Yeah, so today I'm going to present an article accelerating CAR T cell manufacturing. So in this new platform, which is called Fast CAR T, where CAR T can be manufactured only in 1 day, comparative to the conventional CAR T therapy, which takes usually at least a time of 2 weeks, which is really critical for the patient where the disease is very rapidly progressing. So especially as we have seen that CD19 CAR T have shown very successful results in hematological cancers. So these CAR T cells, fast CAR T cells have shown very high expansion rate, younger population, less exhausted populations, and very high potential to eliminate that hematological cancers. So in what they have mentioned in this article in fasty CAR T cells, they have found out that their expansion is very high. They have very high percentage of CD62L positive, CD45 negative, which is for stem cells, memory cells. That means they are less differentiated cells. And that central memory cells is high in these fasty cells. Then again, there is low expressions of PDL PD1, lag3, TIM3, which is for, these are the markers for exhaustion. So that means these cells are less exhausted in compared to the conventional CAR T. Sorry. Again, they found out that the expression of this CXCR4 gene is also very high in these fasty CAR T cells. That means there is that migration towards bone marrow is higher than the normal CAR T cells. So this is the manufacturing layout what they have mentioned. So the normal first day, what they did, they isolate the T cells, which took 2 hours. They've used open system, semi-automated, semi-automated system. So mostly they have used thermo components. So they've isolated T cells by using Dynabeads, which has CD3, CD28 on that. And after isolation, they transduced the cells, added the vector, that viral vector to it, and transduced for 20 hours, where these beads are already attached to it. After that, they've divided it into two parts. One is where they've directly isolated the cells the same day, and in the another one, they expanded the cells for 7 days. So here after activation, they removed removed the beads and then formulated the whatever number is required and then cryopreserve. In this case, the second case, they expand these cells for 7 days. After that, they prepare the formulations and cryopreserve. So sorry. After that, they thaw the cells, expand further for 3 days in GRAC system, and then check their functional analysis. So here what they found, first they checked the viabilities of these cells, isolated cells on day 0. So these CD3 cells, the viability was good. Then after that, they found out the recovery, post formulations recovery, what is the percentage of the CAR T cells. So both in 24-hour process and 7-day process, the percentage of post formulation, the percentage of CAR T cells was almost similar. Then they have found out that what is the that viabilities of these cells in comparison to the total cell count, initially the initial cell count which they have taken after the isolation. So here this is the thawed cells, and that number of these cells is lesser, and then viability is this much. Same in 24 hours post formulations, they have found out that because they are not expanded, the cell number is lesser but viability is very high. And in 7-day post formulation, these cells where they expanded it, that the percentage was high, cell count was high, and the viability was lower than that the 24 hours formulation cells. Then later they check the immunophenotyping of these CAR T cells. So in that they have checked the percentage of all T cells, B cells, monocytes, and NK cells. So these are the leukapheresis cells where they've calculated the percentage of all these T cells, B cells, and NK cells. So this is definitely is high. In isolated cells, these are the percentage of T cells, isolate, and this is the B cells, mononucleoside cells, and then NK cells. So NK cells are also isolated because NK cells has CD3 56, CD3 positive, 56 positive, and they have isolated using CD3, CD28 beads. So that is why NKT cells, we call them NKT cells, they also came in an isolated cells. Then further, after 24 hours of the process, they found out that percentage of T cells was higher. Further, the percentage of other cells was reduced. But in 7-day process, the percentage of other cells like NK cells, NKT cells and all cells was reduced. So here it confirmed that these isolated cells number is good, their viability is also good. Then again, they have found out these cells, is this screen is visible? Can you see these boxes?
Yes, ma'am.
Yeah, okay. So they have found that CAR-T expressing cells, what is the percentage? Because what happens, there is pseudo transduction is also there. So they have found out these are the, yeah. So in this, that pre-cryopreservation, I mean, after 24-hour process, the percentage of cells is 69%. Here they have shown 24 hours percent, that CD19 containing cells, the percentage is pre-cryopreservation is 76. And in 7-day process, they have found out that this CD19 positive cell percentage is lesser. But after thawing and then post thawing 3-day expansion, the cell here also, which was initially 76%, it reduced to 9.7%, and in case of 7-day process, it reduced to, it was almost similar. So that means here they've concluded that this is because of pseudo transduction. So that it here it was showing that because virus was already present there on the membrane, so because of that the percentage of CAR positive cells was high. Then the expansion, fold expansion also they have checked. So pre-cryopreservation we can see because in case of 7-day process cells, they are expanded. So that is why that cell number is higher. And in case of 24 hours, because directly they were cryopreserved, the number of cells was very low. Then after 4-day post-thawing, when they again expanded the cells, these cells, 24-hour cells number increased eventually. Then again CD19 cells, they have checked that what is the cells which carry CD19, that means CD19 positive cells. So pre-cryopreservation, the number here, the correlated to this one, the number is this much, and then here the number is much higher. Then further they have checked the CD4 CD8 ratio. So CD4 CD8 ratio shows the expansion, the efficacy of these CAR T cells. Usually in when the manufacturer they keep this CD4 CD8 ratio should be 1 technically. So initially that number of CD4 CD8 CD4 cells, that means that these cells are that the CD4 cell number is higher. After 24 hours, that still CD4 CD8 ratio is high. In 7-day process, this is equal to 1. After 3 days thawing, this ratio CD4 to CD8 ratio came down lesser than the previous one, and in case it was the same. So these cells, that they've concluded that these cells, fast CAR T cells, they are more expendable. Their cytotoxic, their efficacy is higher than the 7-day processes. Then they have checked the efficacy of these cells by measuring the percentage of stem cells, stem memory cells. So these stem memory cells, these are CD. They checked with the what is there is marker. Let me check that CD62, CD62 for R45, something like that. I exactly forgot. So here they have checked in this the 24 hours that pre-cryopreservation. Here we can see the population of these cells is high, 46%. That means these are more or less younger cells, and they have better stem cells memory in comparison to the 7-day process cell, where these stem cells like characters is only in 14.65% cells. Similarly, in after 3 days process, here we can see this is 59%, and in this case, this is only 12%. So that means these are more undifferentiated cells. That 1-day cells, more undifferentiated cells, and have better stem cells, stem like memory cells is higher. Yeah, CD45 and CRC7. So these are the marker for the memory. So here also they have checked. These are with the let me check. Yeah, memory phenotype. So in this is with the LV positive pre-cryopreservation. So, and this is after 7 days. 7 days and 20. Yeah, so here we can see the percentage of these memory cells is higher in case of 24 hours. 24 hours cells. Here we can see even 3 days post, we can see here that these cells, the number is lesser in comparison to the 24 hours manufactured cells. Then here they have checked for the marker of exhaustion. So PD1, lag3, TAM3, they have used different markers to check. So here again we can see in 24-hour process cells, this expression of these markers, PD1 is very less. Where in comparison to this PD1 is very less, here in case of 7D, this expression of this PD1 is much higher than the 24 hours percentage cells. Then again they found effector to target ratio. Effector to target ratio is like that what percentage of to check the efficacy of this CAR T. That effector is the CAR T cell and the T is the target. So they've checked that also. So that this is showing the percentage of killing. So in percentage of killing, that 24-hour cell, it when this ET ratio is lower, then but that percentage of killing we can see in 7-day process. In 7-day process cell, this killing is much higher in 24-hour cells when they have used different doses of CAR T, like less. Here they have used lesser. This is the baseline and this is the higher. So in all cases we can see that the cells, the killing efficacy of these 24 hours manufactured cells is much higher. Then in cell lines, because they have used that in vitro, in vivo, and then in clinical studies also. So here also they have found out that secretions of IFN, that cytokine secretion. So cytokine secretion also they can see that in 24 hours manufactured cells, the cytokine release is much higher than the 7-day synthesized cells. Then they check the average count that these cells, but definitely because these cells, the number they were expended for longer period of time, so that number was higher. So in this study, they have highlighted that this innovative 24-hour process can increase the production of these as of now they have manufactured only CD19 cells. This shortened manufacture process could improve patient outcome where therapy is required in very urgent basis. So on the basis of this therapy, I would like to present another paper where they have used these kind of cells in clinical trials. So in CD19 CAR T cells for B cell acute lymphoblastic leukemia, this is first kind of human clinical study. So as we know that in case of though CD19 CAR T cells have shown very wonderful effect in treating CD9 this lymphocytic leukemia with remission rate of 70 to 80%. But sometime what happened because of the that progressing disease and the error in manufacturing, that many patients die, and there is data also that 20 to 30% of enrolled patient with the B cell acute leukemia, 20 to 30% died because of not receiving the therapy. So that's why this shortening the time of CAR T manufacturing is really important. So in this the first phase 1 trial study, they have used these fast CAR T cells. So first, the in vitro studies, they have shown that expansion of these cells, fast CAR T cells, is much higher than the conventional CAR T cells. Then again, similar to the previous results, the percentage of these stem cell like character is high in fast CAR T cells comparison to the conventional T cells. Again, that these exhaustion marker also, they have shown that expression of these markers PD1, lag, and then TAM is very much lower than that of the conventional CAR T. Then in vitro studies, they have found out that the killing efficiency is also these fast CAR T cells is much higher than the conventional T cells. They have used two cell lines, NAM6 and then the Raji6, which these two cells are model for B cell leukemias. So here also they have shown that here again they have checked that ET ratio, effector to target ratio. So in case of fast CAR T cells, that efficacy is much higher than the conventional CAR T cells. Then in vivo experiments also, they have used different doses, and after that they have found out that in case of fast CAR T cells, that remission is much higher than that of the conventional CAR T. Then in these mouse model, this and Nog-Raji model, where they have used non-diabetic mice model, where they have transplanted these Raji cells and a xenograft model, and then checked the efficacy of these CAR T cells, different CAR T cells, high dose, different doses of these CAR T cells, and they found out that both the doses that in that fast CAR T are better than the conventional CAR T cells. Then they found that after transplantation, they checked what is the bone marrow translocation of these cells. So in case of our fast CAR T cells, the translocation to the bone marrow is higher than that of the conventional CAR T cells, which shows that they have high infiltration rate and more homing effect, and they are more targeted towards the tumor site. So this is how they enrolled. Their total 20, initially 27 patients were screened. Out of 25 enrolled, and they went for the infusion. These were the dose which were given to these patients. So these were the CRS and CNS can be seen after the infusion of CAR Ts. So in almost every all patients, they have shown grade 1, grade 3 CRS. These are in detail. They have explained that how many patients shows grade 2 or grade 3 CRS. So then after infusion, they check that what is the copy number of the CAR T. So here we can see that it doesn't depend on the doses what they have taken. That the peak we can see that after after 7 days, the peak CAR T number was very high, and it was that in months we can see remain up to the several months inside the body. Then here also we can see that they found out that the CAR T number in the blood. So that up to this is how many months? So up to up to 3-4 months that CAR T was there. Then copy number also, the different doses they have checked, that dose level 4, dose level 3, they have checked that this is the CAR T numbers. And again they have found out that whether immunosuppressive they have given with corticosteroids and non-corticosteroids, still the copy number remains the same. That means that this immunosuppressive environment doesn't hamper the copy number of CAR T, it remains there. So again they found out that in how many patient what is the that effect. So in that's patient after 2 weeks, 75% out of almost all they have disease free remission. And then in another 2 weeks it increases, that if they have this reach to complete remission free negative. So here I would like to, and then this is the post CAR T infusion up to 1,000 days. They have checked the effect of this CAR T. So here we can see in almost 25 patients there is disease-free remission. The 2 patient died. Yes. 2 patient died after because what they did after the infusion of the cells, they did stem cell transfusion. So after that stem cell transfusion, they've checked what is the effect, and then this is the result. So here I would like to conclude that almost this CRS was manageable, with 24% was showing grade 3 CRS and 28% neurotoxicity, predominantly in pediatric patient. And then further they have shown that day 14 out of 25 patient, 23 patient they've reached minimal residual disease with complete remission. Then after that, out of these 25, 20 patients, they went to allogenic hematopoietic stem cells transplantation. Out of these 20 patients, 15 patients were disease-free in 34 days. One patient relapsed, and out of these 24 patients died because of transplant-related mortality. And out of the 3 patients who did not undergo the transplant, they remain remission free until 10 months. So this data indicate that these fast CAR T cells shows preliminary good promising result in early efficacy in B-cell acute lymphoblastic leukemia. So here I can conclude these fast CAR T cells have better expansion capacity, have younger number of cells with less exhaustive phenotypes, better potent cytotoxicity. And in this first clinical trial, they have found out that it can show better result in further clinical trials. There is another study also which is going on related to the same fast CAR T.